inaccessible chromatin.
Thanks, Toshita. So, hi everyone. So I'm Shaun from Penn State. What I want to talk about today is how transcription factors with similar DNA binding domains can nevertheless diversify their binding sites, bind to different sites in the genome, regulate different genes. So there's various strategies by which this could occur. If there are a pair of parallel transcription factors, two transcription factors with similar DNA binding domains, if they don't want to be redundant with one another, they could evolve different DNA binding preferences, intrinsic binding preferences, recognize different motifs, and thereby bind different sites. Similarly, they could also evolve different protein-protein interactions, just like we heard about from Kyle, to form different cooperative or competitive interactions. What I want to talk about today is that another way that they could diversify, which is by evolving different preferences for relatively closed or open chromatin. So if we think about transcription factors binding to relatively closed chromatin, we often think about pioneer factors, and FOX transcription factors are prototypical pioneer factors. So FOXA transcription factors were amongst the first to be recognized to be able to bind to the nucleosome and to thereby open nucleosome and make some regulatory sites competent during development for regulation. You might think that if it's a pioneer factor, it should have some ability to bind to closed chromatin no matter what the context is, but that's not true. We know that FOXA transcription factors bind to different sites in different cell types, so there must be some effect from the cell-specific chromatin environment that's guiding these FOXA factors to different sites. What we don't know about is the rest of the FOX transcription factor family, all these other transcription factors that are encoded in the mammalian genome that have Forkhead binding domains. Are these also pioneer factors? Are they affected by cell-specific chromatin environments? Not much has been done to look at this. So there's about 40 plus transcription factors encoded in the genome with Forkhead binding domains. We're not going to look at all of them today. We're going to look at 5 of them. And specifically, we're going to look at these 5, FOXA1, FOXC1, FOXG1, FOXL2, and FOXP3. They're each involved in different developmental lineages. Each have different roles in development and in some diseases. But as far as we know, they all have quite similar DNA binding preferences, at least in vitro. They all like to bind to this TG-TTTAC motif, at least from what's stored in the SysBP database. So what we're going to do is overexpress each one of these one by one in mouse embryonic stem cells to see what they do in a chromatin environment, not in an in vitro environment. After 24 hours of expression, we're going to measure their binding with ChIP-EXO, and we're going to measure the outcome of that binding with RNA-seq and ATAC-seq. So when we do this, the first thing we see is that there's some sites on the genome where all of these FOX factors that we've looked at bind similarly, but there's many more sites like this where subsets of the factors are binding and other factors are not binding. So there seems to be quite a bit of differential binding overall on the genome. If we just look at all of the sites for each of the factors one by one, here I'm just stacking up the thousands of sites we find from each FOX factor separately. The primary difference that we see amongst these FOX factors is actually the intrinsic binding preference. There are a few different flavors of cognate motif that are recognized by these FOX factors. FOXA1 and FOXG1 seem to prefer this TG-TTTAC motif, which is basically the same as the cognate, the in vitro binding preference. Both of those factors also bind to this almost like a tandem repeat of TG-TTT. Is preferred by FOXG1, also recognized to some degree by FOXA1. The other 3, FOXC1, FOXL2, and FOXP3, bind to this simpler TG-TTT motif, albeit with some T-rich flanks. So given that they're recognizing different cognate motifs, or variation in cognate motifs, you'd expect that there is some differences in where they bind, and that is true. When we look at a clustering of these sites, and by the way, I'm dropping out FOXC1 for the moment because it was too few sites to include in clustering. But you can see from the rest of the factors that are left from this clustering that there's some sites that are recognized that are preferred by FOXA1, some sites that are preferred by G1 and P3, and some sites that are preferred by L2. Remember, this is all in the same chromatin environment, right? We've overexpressed these exactly themselves. First thing we did here was ask if there's any motifs that explain the differences between these different categories of sites. And the answer is not really, compared with what I've already shown you for the cognate motifs. The most enriched motifs in each of these different categories is similar to what I've already shown you. There's no specific motifs that explain the difference, for example, between the sites that are bound by FOXG1 and P3 but not L2 versus these sites which are bound by L2 but not G1 and P3, right? They have the same sort of tandem repeat motif enriched. This is the simplest motif enrichment we've done. We've done other like discriminative motif finders as well and things like that. We're not really finding anything special on the sequence that discriminates between these categories. So what else could be happening here? Well, remember we've overexpressed each of these in mouse ES cells, and we know a lot about the chromatin environment in mouse ES cells. Lots and lots of different experiments from lots of different labs have been done in this. So what we can do here is contextualize the binding sites that we see for these FOX factors at 24 hours with what was happening before we overexpressed the factors in the underlying pre-existing mouse ES cells. So when we do that, we do see some variations, quite a bit of variation in terms of the associations between these different binding sites and what was happening in the pre-existing mouse ES cells. For example, FOXP3, over half of their sites are already accessible or already marked by relatively active histomodifications in the pre-existing mouse ES cells. In contrast, FOXL2, relatively few of its sites are already open or marked by active histomodifications. These are associations here with individual histomodifications. We can compile these, of course, using Jason's ChromaHMM approach here to get chromatin states. The same messages here, though, of course. So FOXG1 and FOXP3 seem to prefer to bind the sites that are already marked as enhancers or promoters in the ES cells. FOXL2 and C1 have a greater preference for sites that were marked as quiescent or just somewhat inert in terms of the histomodifications in the pre-existing ES cells. FOXA1 is in between these different extremes. So I've shown you that there's some differences in sequence preference intrinsically. There's some difference in the preference for association with different histomodifications. How do we put all this together? Well, one thing that we've been doing, and JNU is in the audience here that's been working on this, we're trying to build models to recapitulate the differential binding that we see in the mouse ES cells. And we're trying to do that using neural networks that are either trained with sequence alone. These are convolutional tower neural networks. I can describe more later. So we're either training with sequence alone or with combinations of sequence and pre-existing chromatin. So chromatin modifications, chromatin landscapes that come from the ES cells. So we can compare and contrast these to try and learn what the features are of these various binding sites. The first easiest thing we can do is compare how well they perform. So I'm going to show a very simple metric of performance here, which is the mean squared error. By the way, this is a regression setting. We're trying to predict the chip enrichment for these various factors at the various sites. You can see that adding chromatin into the model here, this is the sequence plus chromatin model in orange, improves the performance. So mean squared error, the lower you are, the better. So it improves the performance quite a lot for FOXP3 and for FOXG1, but less so for FOXL2, suggesting that perhaps the chromatin features don't contain that much information that's informative for predicting FOXL2 binding, whereas it does for FOXP3. We can go a bit further here, though, and see if we can predict the types of differential binding that we're showing in the ChipExo here. So if we train a sequence-only neural network, you can see that we're not doing very well in recapitulating the differential binding at sites that are bound by one or another of these FOX factors. I mean, if you squint, you can see that the FOXA1, it at least is getting a bit higher prediction here in the sites that are bound by FOXA1 or preferred by FOXA1. And similarly for FOXL2, you get a little bit higher in predicted enrichment at the L1 preferred sites, but not much. If you include chromatin information, you're now seeing a bit more performance. Again, it's not great still, right? We're still working on this. But we see a little bit better performance for the FOXP3 and FOXG1 at the sites that those factors like to bind. It's finally, now that you're giving it chromatin information, it's recognizing that these sites should be preferred by FOXG1 and P3. We actually see a bit of a decay in performance, though, at those same sites for A1 and L2, suggesting that either the networks learned the wrong information or that we're missing something from our chromatin environment in our model. So we're still working on this, but we can go into these models and ask what types of features they're recognizing when they're making these various predictions. We can do that with deep SHAP feature attribution approaches. So when we do that for the sequence plus chromatin model, and just project the sequence feature attributions onto the same sites, we can see that there's not that much difference, especially in FOXG1 versus FOXL2, which bind quite differently on the genome. The networks are basically recognizing the same sequence features at these sites. It's only when you start looking at the feature attribution for the chromatin tracks, like, for example, the attack-seq, that you start seeing differences across these factors. So, for example, the network learns that the lack of accessibility at these sites is a strongly negative feature for FOXP3, just as an example here, whereas not a negative feature for FOXL2. So as I said, we're still working on this, but we're hoping that this gives us an approach to start deconvolving these different types of features that predict binding of these various paralogous factors. Excuse me. So one last thing you might be wondering is what happens to these sites after the FOX factors bind? Do they increase in accessibility as you would expect for pioneer factors? So we're looking at this question by, first of all, splitting out the sites that were already accessible in ES cells versus the sites that were inaccessible or relatively inactive in ES cells. This is not just based on attack-seq, but also based on the chromatin state overlaps, like active would be counted as enhancers, promoters, etc. At either set of sites, you do see, at either set of sites, you do see an increase, a strong increase in accessibility. I'm sorry, I keep hitting the mouse here. You see a strong increase in accessibility at FOXA1 sites, regardless of whether those sites were already active or inactive in ES cells. You also see some degree of increase in accessibility at FOXL2 and C1 sites, but not so much at the sites that were inactive before or inaccessible before overexpression of these factors. And you see very little, if any, increase in accessibility at G1 and P3 sites. In fact, you see an increase in accessibility, but that increase was also going to happen anyway, even without these factors being expressed. And we can see that by looking at a null control line where we overexpress nothing, just let the ES cells differentiate for 24 hours. And the accessibility we see there is in the red lines here. You can see that that's mostly indistinguishable from what happens in the context of the overexpression of the FOX factors. So in summary here, we think that this is another strategy we should be considering when we're thinking about how transcription factors, paralogous transcription factors, can diversify their binding sites and their regulatory programs during development. We've seen here for these FOX factors that they can change their sequence preference or their chromatin binding preference. This is just to note the second time that we've seen this for a family of paralogous transcription factors. We have seen a similar story with HOX transcription factors, where we recognize one HOX transcription factor in particular, HOXC9, seems to have this different ability to break into relatively closed chromatin. We published that collaboratively a couple of years ago. Just to thank everyone that's involved in the work here. So JNU is here, as I said. Sunny Aurora worked on this also in my lab. This is the collaboration with Frank Pugh's lab in Cornell and Minoru Ko's lab in Keele University. Thanks so much.
Hi, really nice work. I noticed that when you added the chromatin in, your prediction of the peak intensity in the middle seemed to get broad. Is that something to do with the way, like, the prediction was sharper, I thought, in the sequence only, and it became broader when you added the chromatin in?
Yeah.
Is that something about the way the chromatin model is encoded, or is it actually losing the sharpness of the prediction somehow?
I mean, one thing, and this is not your question, I know, but one thing I didn't clarify was that it's broader than the ChIP-EXO signal because we're making predictions in 500 base pairs. But yes, what you're noticing is the spread of high prediction outside of the core region. And I think that's just because it's learning in association with chromatin, and the chromatin features are spread out from the ChIP-seq resolution. And so it's thinking that those sites, it's increasing the prediction of those sites, even if they don't necessarily contain strong motifs. Yeah, thanks though, good question.
Thank you, very interesting talk. I'm just wondering if you have looked at the other transcription factors, so-called, right? Are differentially enriched in different classes of these fork transcription factors.
So other ES transcription
other factors that determine or that enrich.
Yeah. But we do see overlap with transcription factors that are expressed in ES cells, but no specific enrichment beyond what we would expect from the enrichment with enhancers or promoters, right? So there's no pairing of a transcription factor. There's no factor we could see in ES cells, and we looked at dozens and dozens of characters in mouse cells. There's no specific one that would explain the binding of one or another of these factors. That's definitely something we look for, though. Yeah.
Thank you.
Thank you.
So I'm sorry if I missed this, but I was wondering how long your input sequences are that you give to the model, because there have been a number of studies showing that sequence can actually be predictive of chromatin, and for histone modifications, you also need something on the order of thousands of base pairs instead of hundreds. And if you considered maybe trying a longer sequence to see if that might improve the sequence-only model, because it's also telling you something about the chromatin.
Yeah, our inputs are 500 base pairs. I don't think we've varied that too much in our optimization. It's a good question. We should look a bit broader at that.
Thanks.
Thanks.
Excellent. So that's